Okay, thank you, Justin, for the introduction, and thanks for the opportunity. My name is Mehdi Razavi. I'm an associate professor of medicine, and also I'm a member of Bionics cluster with secondary joint appointment with Department of Material Science and Engineering, and also biomedical engineering program. My research is always focused on an area that brings together advancements of material science with medicine. What we do is called nanomedicine. We synthesize nanoparticles or nanobubbles that you can inject them inside your body. They can move throughout your bloodstream and kind of house-to-house search or cell-to-cell search to find effective genes into the cells and then deliver therapeutics into the cells. So our goal is to use them for treatment and prevention of osteoporosis. The other area of research is on regenerative medicine. We build bioactive materials that can regenerate or reconstruct damaged tissues. So our goal is for bone repair and regeneration because of osteoporotic fractures. So these are two subjects that I'm going to discuss today. My lab is located at Burnett School of Biomedical Sciences. The focus is on biomaterials and nanomedicine. So the first topic is a targeted ultrasound responsive nanobubble-based siRNA delivery system for osteoporosis. 1 in 3 women and up to 1 in 5 men at age of 50 or older breaks a bone because of osteoporosis. So this is responsible for 8.9 million fractures every year, or one fracture every 3 seconds, and is expected to significantly increase by 2050. There are medications available, including the use of bisphosphonate, but side effects are jaw osteonecrosis and gastrointestinal issue. So we think that there is a better way for treatment of osteoporosis using nanomedicine and also using nanomedicine for gene therapy. So gene therapy, as you know, is a very important aspect of healthcare, which has stalled over the last several decades because of challenges that we have faced for delivering genes to the right place in your body. One thing is how to fix a cell using gene repair, and the other important thing is how to deliver those genes to the right place in your body. And these challenges have started to be solved. There are new delivery techniques based on nanotechnology that you could use to inject nanoparticles into the bloodstream. They can then search to find cells of interest. In our case, is bone cells, and then they can deliver their content into the cells through a different mechanisms. In our case, we are interested in using ultrasound responsive nanoparticles. We apply ultrasound to nanoparticles. They are collapsed, and then they can deliver therapeutics into the bone. Ultrasound alone can also enhance bone formation. These nanobubbles are synthesized using a sonochemical technique and is characterized through a series of experiments, including transmission electron microscopy, in vitro and in vivo testing. Size of these nanobubbles ranges from several tens of nanometer to around 100 or 200 nanometer. So they can leave the vasculature in case of smaller nanobubbles, or they can remain in the vasculature in case of larger nanobubbles and then target vascular endothelial receptors. Phase changes are also possible from liquid to gas using ultrasound exposure. These nanobubbles encapsulate siRNA or a small interferon RNA for gene silencing of osteoporotic causing genes. And then we also look at their stability over the time and sensitivity of these nanobubbles with ultrasound. You can do cell culture these nanoparticles with cells. For example, osteoclast. In our case, osteoclast is bone resorbing cell. So we want to see if these nanoparticles are internalized into the cells and they can deliver their siRNA into the cells. We also look at function of osteoclast in terms of TRAP activity and to see if these nanoparticles are able to reduce TRAP activity and bone resorption activity of these osteoclasts. So nanomedicine has been around for a significant period of time, but what's evolved over the last decade is targeting. So instead of having them to just wander around the bloodstream, how do you actually get them to target cells? and then targeting to not only giving you information about a disease, but also to deliver therapeutics into the cells. So we look at bone targeting and biodistribution of these nanobubbles using overreactomized mice model of osteoporosis, injected nanoparticles systemically, and then we see if these nanoparticles can go into the femur, tibia, and spine, sensitive to osteoporotic fractures. So we have seen their bone targeting potential, but we have also seen some of these nanoparticles are accumulated into other organs, including liver, spleen. So our goal is to enhance the bone targeting of these nanoparticles and also reduce off-targeting by modification of physicochemical characteristics of our nanobubbles, including their size, their monodispersity that we are exploring using advanced manufacturing techniques. You can also do in vivo, test in vivo effectiveness of these nanobubbles after systemic administration. These are bone histology images using H&E mesenterochrom and cathepsin K staining. We are interested in cathepsin K because we are using cathepsin K siRNA, and we want to see if there is any downregulation in cathepsin K expression. Cathepsin K is one of the important osteoporotic causing genes, but there are also other genes that we can target in future. So using these data and also quantification, we look at increase in cortical thickness, also decrease in trabecular spacing, increase in trabecular thickness, and also reduction in cathepsin K expression. So physics of understanding of stimulation of these nanobubbles, interaction with ultrasound, their nucleation, formation, bone targeting have been well studied over the last few years, and we now have a pretty good understanding of how to synthesize, characterize, and also optimize these nanobubbles to get to a good ultrasound responsiveness for siRNA release as well as bone targeting. Our future research in this area is focused on improving bone targeting through modification of physicochemical characteristics of these nanobubbles, and also using the optimal dosage of these nanobubbles and also cathepsin K encapsulation in siRNA to achieve the best effectiveness in terms of reduction in bone resorption for osteoporosis. My second subject I wanted to discuss today is bioactive magnesium-based nanocomposite for bone regeneration. And we've been supporting a vision for well over a decade now, essentially through targeting two main goals: controlling biodegradation and also improving bioactivity of magnesium-based materials. The goal here is not really to develop new magnesium alloy, is to focus on solving important challenges of magnesium materials that have limited their clinical application, which was corrosion and low bioactivity. So we are synthesizing nanoparticles based on bioactive glass ceramic, able to release metallic ions, including calcium, silicon, and magnesium, culturing them with macrophages, looking at macrophage polarization, and we want to see if these nanoparticles can have control over immune response through reduction of tumor necrosis factor alpha expression, as you can see in the figure, and also increase in pro-healing cytokine expression such as interleukin-1 RA. And these nanoparticles are incorporated into the magnesium alloy, and because they have, as you know, nanoparticles have a high surface area, so they always tend to aggregate. So we want to deaggregate and uniformly disperse them into the magnesium alloy. So we have developed new advanced manufacturing technique for microstructure refinement, for elimination of a structural defect, as well as improvement of dispersity and deagglomeration of these nanoparticles. And then these materials are implanted into the femoral defects of rats. We look at integration, osseointegration with surrounding tissue. We look at structural integrity. We look at hydrogen bubble release, because hydrogen release is one of the side effects of using magnesium because of their fast corrosion. And also we do micro-CT imaging in order to evaluate new bone formation, as well as other testing and assessment like histology using H&E mesenterochrom. Here, for example, black arrow shows implant location, red arrow is fibrotic body response, and green arrow shows a new bone formation. So our future research in magnesium field is focused on controlling corrosion through advanced manufacturing and microstructure refinement, understanding the corrosion mechanism, not only in vitro but also in vivo animal models, and also really understanding osteoporotic bone regeneration mechanism using these two types of materials. I would like to thank our sponsor, NSF, for supporting this research. A special thank to my team working really hard to collect these data, our collaborators at Bionis Cluster, BSBS, CECS, Nemours Children's Hospital, and CREAL. And thank you for this opportunity. Thank you for listening.